So I'm going to go through glycolysis and basically overall each of the 3 types of catabolism that we went through in class. So remember, catabolism is a type of metabolism, and you break down, or it's basically breakdown of sugar, like glucose, to produce energy, right, as ATP. So there's 3 types of catabolism that we talked about that are in microbes, and the first one we talked about was aerobic respiration. And the other type that we talked about was anaerobic respiration. And then the last one was fermentation. So these are 3 different ways that we do catabolism, so how we produce energy in each of these 3 pathways. Okay. So the first one, we went through the worksheet that you have, is aerobic respiration, which we separated into 3 parts. But basically, for all of these 3 pathways to start producing energy, all of them will involve glycolysis. So this one starts with glycolysis. This one starts with glycolysis. And this one starts with glycolysis. So regardless of if there's oxygen or whatever their requirements are for each of these pathways, glycolysis is the start of all of these pathways. And so what is glycolysis? It's when we break down glucose and we change it into pyruvate. And we worked through this during class, but I'm going to show it right here. So again, we have 3 catabolic pathways. And these 3 pathways, one of them requires oxygen. This one requires nitrate or sulfate. Okay, so nitrate or sulfate are the things that this one requires, and this one requires nothing. This happens all the time, right? All it requires is that, it's basically the default. So fermentation is our default kind of catabolic pathway that almost always happens. If there's no oxygen and if there's no nitrate, then the bacteria will choose fermentation. It's a very simple pathway. So let's go through glycolysis, since all 3 of these pathways are going to use it first, regardless of oxygen or anything else. Okay. All right. So this is glycolysis. And this is phase 1. We call this phase the energy investment phase. Okay, so first we start with carbons for glucose. So we have carbon 1, 2, 3, 4, 5, and 6. And this is our glucose. Oops. Okay, and then next we go from there. So this is how I want you on the exam to draw the glucose, again with the oxygen on top right here. Okay, there's the oxygen on top, and then you have your 6 carbons labeled with their numbers. Okay, so from here we're going to draw glucose again and all the carbons with it. This is kind of like a hexagon with a little carbon sticking out, so we got a hexagon ring here. And then we label the carbons again, and then we have investment of energy. So remember, ATP has 3 phosphates. You could write it as APPP. And remember, it's like money, right? ATP is like energy investment. It's like having $3. 3 phosphate is $3. So if I use one of these phosphates and transfer it to that carbon, then I'm going to change ATP into ADP. So now I get let me erase there fast. So now we get APP, right? Also known as ADP. So ATP transfers a phosphate right here to carbon number 6, and then therefore ATP becomes ADP. And let me just draw that arrow again. So there we go. We have our first step of glycolysis, and now we call this molecule glucose 6-phosphate. Okay. All right, so the next step here is we're going to now rearrange this glucose molecule here into fructose. So that's the third step. So we're going to keep this this phosphate right here, but we're going to just rearrange this whole ring of carbons. So how I like to remember is fructose is like a hexagon. So we draw the oxygen first. Then the carbons, now there's 4 in the ring instead of 5 in the ring. So and then carbon 1 went up like that. Okay, so there's 6 carbons. It's just all the carbons got rearranged. Carbon 1, 2, 3, carbon 4, 5, and 6. And we call this fructose. Okay, so it's fructose 6-phosphate. Okay. And now here we're going to add a phosphate right up here because it was already there over here. So now we kept the phosphate bonded to the carbon. Okay, so we have fructose 6-phosphate. Now the next step here, I'm going to go down, is we're going to change fructose 6-phosphate into fructose 1,6-phosphate. So we're going to invest energy one more time. So let me just draw the fructose again. And then you're going to go up like that. Carbon 1, 2, 3, carbon 4, 5, 6. Same like you did on top. And then I'm going to invest another ATP. We're going to we could write APPP. And then we're going to we already had a phosphate over here from up here. Okay, and then we already have a phosphate right there. So we're going to invest another ATP. Going to transfer a phosphate right there, and that's going to make ATP into ADP again. So it lost one of its phosphates. It invested one of its dollars, right? Just like money. So now it's just APP, or also known as ADP. Okay. All right. So this one is called fructose 1,6-bisphosphate. All right, so this is the part where now we used 2 ATPs here in this whole energy investment phase. We used ATP. So this is phase 1 of glycolysis. So all of this is glycolysis, regardless of like the phases. It's all still glycolysis. It's just glycolysis has 2 phases. So now we're going to start the energy production phase, or the energy conservation phase. Was phase 2. So this is the start of phase 2. So let me draw. This is phase 1, and then now we're going to go into phase 2. This is also called the energy conservation phase. Conservation means production of energy. So from this phase now, we're going to keep going with this fructose, and we're going to split this ring up. So actually, let me do this one, and this one. So carbon 1, 2, 3. So we're going to split these 3 carbons from these 3 carbons here. We're going to split these into 2 groups, okay? So we have carbon 1, 2, 3, and carbon 4, 5, and 6. So I know this is 4, 5, 6 right here, but we need to rename that to carbon basically 1, 1, 2, and 3, because 4 is smaller, right? So you're going from small number to large. 4, 5, 6 changes to 1, 2, 3, because now we made 2 separate molecules here. So we're going to change these ones to carbon 1. Oh, yeah, carbon, sorry, 3, 2, and 1, right? So we're gonna, I'm gonna erase this one so that you know now this is carbon 1 here at the bottom, carbon 2, and carbon 3. Okay, so now when we do that, we have to keep the phosphates that we already had before right here. So over here, I didn't even have them here. We had phosphate here. We had a phosphate over here on carbon number 6. That's why it's fructose 1,6-bisphosphate. And then we have, so it would be carbon 1 on here, and then carbon, the 6 over here is carbon 3 over here. So that's phosphate on carbon 3. So see how this is the same carbon as this carbon over here? I just renamed it to number 3. Okay, so now we have 2 separate molecules when we split up this ring right here into 2 carbon chains. Okay, so the main thing that you see the difference of right here is that right here, these is on carbon, phosphates on carbon number 3 here, and over here, phosphates on carbon number 1. And we actually want these 2 molecules to be the same. So you do have to remember the names for these 2 molecules when they split. So the bottom one is called dihydroxy acetone. And then the top one here is called glyceraldehyde 3-phosphate. Okay, so that's glyceraldehyde 3-phosphate, because the carbon, the phosphates on carbon number 3 here, right? The phosphate's on carbon number 3, so that's why it's glyceraldehyde 3-phosphate. And then down here is just dihydroxyacetone because the carbon's on number 1. So what we want to do now is we actually want to make these 2 molecules the same, but they're different right now. So how do we make them the same? There's an enzyme that comes in and moves this phosphate to carbon number 3. So now I can change this 3 carbons, draw it again, and we have carbon 1, 2, 3, and now I'll put the phosphate on carbon number 3. And when I do that, now I have the carbons here. This one is on phosphate number 3, and this one's on phosphate number 3. Okay, I mean carbon number 3. So now I have 2, this became a different molecule, and now we have 2 glyceraldehyde 3-phosphate. So we have one here, right, and then one right here. And I'm going to highlight these carbon 3 with their phosphates because they're kind of on opposite sides right now. So you can kind of write this again, like flip it over 1. So 1, so it matches the bottom. 1, 2, 3 is basically, it's the same molecule. So I'm going to just redraw it like that. So as long as the phosphate's on the correct carbon, that's all that matters, right? So in this case, if it's glyceraldehyde 3-phosphate, that means it has a phosphate on carbon number 3. Okay, so after this, then we're actually going to integrate. We're going to put in NAD+. So NAD+ comes in. So here comes NAD plus with an enzyme. And it does the same thing to both of these molecules. Oops, I'm going to put it a little further back. So NAD plus comes in. And there's a hydrogen on carbon number 1 that really needs to be transferred to NAD plus in both of these molecules. So once that happens, NAD plus can becomes NADH because it accepted that hydrogen from that carbon number 1. And then when that happens, basically now you have the space, you have like space right here on carbon number 1 to add a phosphate, another phosphate on carbon number 1, which is what happens. So we make 2 NADH here, and then I'm going to rewrite our carbons. Okay, so I had a phosphate on carbon number 3. And now because I made space right here on carbon number 1's, because I removed that hydrogen and transferred it to NAD+, now I have space for another phosphate on carbon number 1. Okay, so I have carbon 1, 2, 3, 1, 2, 3. And you don't have to know the names for these ones. I just basically want you to know that it has 2 phosphates. From here, it gets pretty simple. Basically now we're going to remove all 4 of these phosphates and transfer all 4 phosphates to 4 separate ADP molecules to make 4 ATP molecules. Okay, so you don't have to know like the steps for this one, but there's several like 3 more steps. But really you're just going to bring in now a bunch of ADPs. 4 ADPs come in. Right, they're coming to collect their phosphates, collecting their money from the 3 carbons, the carbon 1 and carbon 3. And so phosphate's going to transfer to ADP, ADP, and when it transfers to ADP, what do we make? We change that to ATP. Now there's 3 phosphates on there. Get more money. So how many phosphates, how many energy molecules did we make? How many ATPs did we make in phase 2? We made 1, 2, 3, 4. So 4 ATP were made in phase 2 of glycolysis. And now when we remove all of these phosphates, I'm going to like move down here. We remove all of these phosphates, what are we left with? We have 3 carbons each. Carbon 1, 1, 2, 3. Carbon 1, 2, 3. And they don't have any more phosphates, right? Their phosphates are gone because they went to ATP over here. So these are pyruvate. Yay! They are pyruvate. So we make 2 pyruvate at the end of glycolysis. So overall, the products that we made 2 pyruvate, we made 2 NADH, and we made 2 net ATP. Okay? Because we used 2 at the beginning, we used 2 ATP and became ATP, right? ADP here. And then we made 4 here. So because we had to use 2 of them at the beginning over here, we had to use 2 of these. Really the net amount is these 2 ATP, right? So 2 net ATP. That's it. So if we go back up here, this process of glycolysis, both phase 1 and phase 2, are involved in all 3 of these catabolisms. Okay, so make sure then after this, it's going to be the Krebs cycle or the citric acid cycle in aerobic respiration, and then after that, it's going to be the electron transport chain, which we went through for aerobic respiration. And then again, the only difference that I want you to know for anaerobic respiration is that a it has a different electron acceptor, which is nitrate or sulfate. So nitrate is for E. coli. It uses that as a final electron acceptor. So, yep, that's it.